Good afternoon, everyone. My name is Amanda, and today I'm going to talk to you about seaweed biodiversity. Seaweed are, simply speaking, marine algae that are macroscopic. This means that you can see them without a microscope and that they live in the ocean. You can also refer to seaweeds as marine macroalgae. They're photosynthetic, and along with microalgae and cyanobacteria, they produce 70% of the oxygen that we breathe in the atmosphere. There are three main types of marine macroalgae: reds, browns, and green algae. And as you can see, they're extraordinarily beautiful. But for scientists who are interested in species diversity, there are a few characteristics that seaweeds share that make them especially tricky to study. Before I dive into these, though, an important question to answer is, how do scientists decide what is a species? How do they group living things into entities that we call species? There are a couple of different concepts that come into play here. And one of the most well-known species concepts was summarized by evolutionary biologist Ernst Mayr in 1942. Species are groups of interbreeding natural populations that are reproductively isolated from other such groups. This idea is known as the biological species concept, and the key to this concept is that generally, the entities that we call species are unable to interbreed. But this can be hard to test in nature, so scientists often use other species concepts as a proxy for the biological species concept. And historically, this has meant using a species concept based on morphology. The main assumption is that a group of individuals that make up a species will have a shared set of morphological characters that distinguish them from other related species. In practice, this is how almost all species identification has been done throughout human history. Even someone with a minimal interest in botany can tell the difference between an oak tree and a maple tree just by looking at the shape of their leaves. But what we have to keep in mind is that just because we can't see a difference between two different groups of organisms with our eyes doesn't mean that they aren't different species. Basically, morphological distinctiveness is not always a good stand-in for reproductive isolation. And now that you understand a bit more about how species are defined and how they've historically been recognized, I can explain why it's sometimes really tricky to delineate seaweed species and what tools we can use to help. The first problem is that seaweeds can have really simple morphologies. They lack roots, stems, and flowers that you see in land plants, for example. This means that sometimes there are very few characters that can actually be used to try and separate different seaweed species. Another cause of difficulties can be convergent evolution. Convergent evolution is when organisms that are not closely related but live in similar habitats look alike, such as the two species shown here. And yet another problem for taxonomists is phenotypic plasticity. Phenotypic plasticity is when members of a species look different when they live in different habitats. The three individuals shown here are the same species collected from different environments. So, simple morphology and convergent evolution can result in an underestimation of species diversity, while phenotypic plasticity can result in an overestimation. One solution is to incorporate other tools to help, such as those that rely on genetic information. One of these tools is called DNA barcoding and uses a short, standardized section of DNA in order to identify species. The idea is to make identification as easy as scanning a barcode at the grocery store. The differences between DNA sequences between individuals can be used to delineate species. These sequences do not always correspond to the morphological characters used by taxonomists, but the assumption is that they do correspond to reproductive isolation. And using a combination of both genetic and morphological evidence can reveal overlooked biodiversity and clarify species complexes that have frustrated taxonomists for decades. It can also be used to find unexpected introduced species and to discover new species. So just to finish up with a few examples, first, a routine DNA barcode survey was critical for detecting the first Canadian record of this invasive species, Dasyphonia japonica, in Nova Scotia in 2012. In 2017, in British Columbia, DNA barcoding was also instrumental in uncovering hidden species diversity within the species Polycyphonia pacifica. Genetic analyses showed that there are multiple genetic species groups that need to be explored and described. And finally, these are two new Canadian species that I described in 2016 with the help of DNA barcoding and morphological assessments. Thank you for listening to my talk.